Learning objective number 2 is now looking inside the ear and specifically at the receptors for equilibrium, which are located in the vestibule and the semicircular canals. But before we get to the receptors, we need a little bit of background on equilibrium. And so within the body, equilibrium refers to a state of balance and stability maintained by the vestibular system. The vestibular system, which can also be referred to as the vestibular apparatus, is made up of the vestibule and the semicircular canals, and they detect changes in head position and body movement. The receptors in the vestibular system send signals to the brain to help coordinate movement, maintain posture, and stabilize gaze. Any disruption to these receptors can result in vestibular disorders or motion sickness, as well as feelings of dizziness, vertigo, or imbalance. So the vestibule is this middle portion of the inner ear here, right between our semicircular canals and our cochlea. So the vestibule is made up of our bony labyrinth. Within that bony labyrinth, we have the utricle and our saccule. So these make up part of our membranous labyrinth. Now, our utricle and our saccule are small fluid-filled sacs, which are lined with sensory hair cells that detect linear acceleration and also changes in the position of your head. So the vestibule, which as we now know is made up of the utricle and the saccule, contain the receptors for what we call static equilibrium. Static equilibrium refers to maintaining the position of the body relative to the force of gravity. So body movements that stimulate the receptors for static equilibrium include tilting the head, so that might be looking down at your lap, as well as linear acceleration and deceleration. Examples of when static equilibrium is required is when you're moving up and down in an elevator, or you're in a car that is speeding up or slowing down. As you can tell from those examples, static equilibrium is most often required when the body is static or at rest or in a static position, and is essential for activities like standing upright, for sitting, and maintaining a steady gaze. The utricle within our vestibule primarily detects horizontal movements and changes in head orientation. The saccule is more sensitive to vertical movements. Now, our semicircular canals are these parts here. So again, our semicircular canals make up part of our bony labyrinth. Our semicircular ducts are these pink parts which sit within the canals and are part of our membranous labyrinth. The semicircular ducts contain the receptors for dynamic equilibrium. So our semicircular ducts contain our receptors for dynamic equilibrium, and dynamic equilibrium is the maintenance of body position in response to movement when there is rotation around an axis or a pivot point. So this can include shaking your head as if you're trying to indicate no, spinning around in a circle, or going on something like a merry-go-round. Each of the 3 semicircular canals, or ducts, detects rotation in a different plane. So our horizontal canal and duct detects rotation around the vertical axis. So this could be shaking your head side to side. The anterior and posterior canals detect rotation around the sagittal and the coronal planes. So this is nodding your head like you're saying yes, or tilting it to one side. Now, taking a close look at the specific receptors, and we're looking at static equilibrium here. And within the utricle and the saccule in the vestibule, which remember are within that membranous labyrinth of the inner ear, are thickened regions called the macula. So up here in this little image, the brown is our bony labyrinth. Within the vestibule in our membranous labyrinth, we have our utricle and our saccule in blue. Within these two structures, we have thickened regions, which we call the macula. So you can see the spelling of macula there. Now, our macula consists of two kinds of cells: hair cells, which are the sensory receptors, and supporting cells, which surround the hair cells. So our supporting cells are this beigey pink colour down the bottom. Our hair cells are in blue. Sitting on top of the hair cells are stereocilia, which are like really fine little hairs extending out the top, and then one long hair cell called a kinocilium. Together, our stereocilia and our kinocilium are called a hair bundle. So we can see that better in this image down here. We have our supporting cells, we have our hair cells, we have our stereocilia, and then there's one big long kinocilium, and together these are called our hair bundle. Now, these supporting cells secrete a thick gelatinous glycoprotein called the otolithic membrane, and this rests on top of the hair cells and surrounding the hair bundle. On top of this otolithic membrane is a layer of calcium carbonate crystals, which are called otoliths. So sitting within the macula, which sit within our utricle and our saccule, are our supporting cells, and then our hair cells. Sitting on top of and surrounding those hair bundles and hair cells is our otolithic membrane. Sitting on top of that membrane is our otoliths, so those crystals. Now, if you, for example, tilt your head forward, so have a look at your lap, or if you're in a car and it accelerates, these otoliths will fall forward or they'll lag behind. And when they do this, they drag that otolithic membrane with them. When that otolithic membrane moves, it bends those hair cells within the hair bundle. And depending on the direction of the bending, will either cause depolarization or hyperpolarization of those hair cells, which can also be classified as mechanically gated ion channels. So when the hair cells bend one way, it will cause depolarization. When they bend the other way, it will cause hyperpolarization. And this is what allows us to differentiate movement in different positions or different directions. Now, in our three semicircular canals are our three semicircular ducts, which make up part of our membranous labyrinth. And at the bottom of each duct is a thickened or dilated region, which we call the ampulla. Within the ampulla is a small elevation called the crista. Now, similar to the macula, each crista contains a bundle of hair cells. It contains supporting cells, and extending out of those hair cells, we have stereocilia and kinocilium, which again, combined, are called the hair bundle. So our beigey pink are our supporting cells, the blue again are our hair cells, and you can see our hair bundles sticking out here. Now, covering the crista is a mass of gelatinous material called the cupula. The hair bundles will extend into that cupula. Also within this semicircular duct is endolymph, which remember is a fluid. So if we're spinning around in a circle or we're shaking our head no, this endolymph or the fluid in our semicircular duct is going to move, but it's going to lag behind a bit. This will drag the cupula with it, which bends these hair cells, which again causes either depolarization or hyperpolarization depending on the way that that cupula moves. When we have the depolarization or the hyperpolarization of these hair cells, it generates a receptor potential. This then travels along the vestibular branch of our vestibular cochlear nerve or our cranial nerve 8. So a bit of a recap. The vestibular and semicircular canals in the inner ear house the receptors for equilibrium. The vestibule, which contains the utricle and the saccule, monitor static equilibrium. So this is activated when you tilt your head or you're moving in a linear fashion or a straight line. The semicircular canals and the semicircular ducts within those canals monitor dynamic equilibrium. And this is activated by movement in a rotational fashion. Looking at static equilibrium specifically, and within the bony labyrinth of the vestibule are the saccule and the utricle. Within the walls of the saccule and the utricle are thickened regions called macula. The macula contain hair cells and supporting cells. Sitting on top of those hair cells is the otolithic membrane. Sitting on top of the otolithic membrane is the otoliths. Tilting the head or accelerating or decelerating causes those otoliths to move or fall forward or back. This movement drags the otolithic membrane, bends the hair cells, and causes a receptor potential. For dynamic equilibrium, and our semicircular ducts sit within the bony semicircular canals. At the base of each duct is an enlarged or a thickened region called the ampulla. Within each ampulla is a small elevation called the crista. Extending out of the crista are also hair cells, which are surrounded by that gelatinous cupula. As the head moves, so do the hairs and the cupula, but the endolymph or the fluid in the semicircular ducts will lag. This causes the movement of the cupula, the bending of the hair cells, and the generation of another receptor potential. For both our static and our dynamic equilibrium, this receptor potential will move along the vestibular portion of our vestibulocochlear nerve and then eventually back to the brain.